Within the microbe population living in our intestines, our gut microbiota, smaller ecosystems of microbes, known as microbial guilds, exist. A guild is dedicated to a specific metabolic process. Our research applies graph theory principles to model the interactions between the microbes within such microbial guilds. Within each metabolic microbial guild, a starting species produces a signal for the next species, and so on, eventually reaching the final species that produces the guild's end product. Each microbial species is represented by a dot or node, and the signaling connections between the species are shown as lines or edges. Each microbial signaling configuration can be represented by a simple path. All possible simple paths starting from node 1 going to target node n are generated. The number of paths increases rapidly. Over 2 million are possible with only 7 nodes. Path density is also calculated. Heat maps display the landscapes of paths with respect to density and path length. Finally, we calculated the average density weighted by the number of paths. The weighted average density decreases as the number of nodes increases, so fewer interactions and hence less energy are needed with higher species diversity to still reach the maximum number of paths. Our simulations show that increasing species richness leads to higher efficiency and resilience within the microbial guild, a starting point for rational microbiota intervention.